This is a production of Cornell University.
No, it's great to be here. And today I'm going to tell you a little bit about the work that my lab does on plant ecology and public health. So the mission of my group is to quantify plant-human interactions to inform decision-making and ultimately to improve public health. To do so, we seek to answer questions including how can we measure, model, and scale up processes that are health relevant. We also seek to answer what are the public health consequences of plants and who is most affected by them. And ultimately, we seek to use this information and to leverage it to inform decisions that will improve human health. So there's a lot of different ways in which plants interact with people from a health perspective. In our day-to-day lives, we may benefit from the cooling effects of trees on a hot day, or from the more subtle but still important removal of air pollutants like particulate matter by urban plants. These plants also have all sorts of other effects too, everything from mental health benefits to hydrological effects like that can reduce flooding in urban areas. Of course, these plants also have negative effects too, such as the production of allergenic pollen or the production of volatile organic compounds, which contribute to ozone formation. Today, I'm mostly going to tell you about one of these in particular, which is allergenic pollen. So I'm going to go a little bit into why this is important and what we're doing about it, and then I'll talk about some other stuff that I do too as well. So most of what we know about airborne pollen comes from empirical measurements taken across the United States by the sparse monitoring network. To give you a sense of how this works, I'm going to zoom into the Finger Lakes region. So we are over here in Ithaca, and the nearest of these pollen monitoring stations to us is up in Rochester, which is about 80 miles away from here. The way it works is there's a sampler that looks kind of like this on top of the roof of a single building up there. And this sampler draws in air that has pollen and fungal spores and other things like that in it. And then once a day, a technician goes up to that rooftop, takes this sample out of the device, manually identifies the pollen grains, and then reports that information to the media. Now, what does this mean if you live in, say, Fall Creek? Well, there's a few pieces to this. Unfortunately, what was measured in Rochester in the morning, that was those measurements were taken over the previous 24-hour periods. So you're talking about yesterday's data. Also, pollen concentrations vary by orders of magnitude over spatial scales of hundreds of meters. And so what's happening 100 kilometers away isn't going to give you very useful information about what is going to happen today for you here in Ithaca. And so without that information, you may not be able to make behavioral decisions like deciding whether you're going for a run that day or whether you should close your windows. And it also doesn't really tell you whether you should take your allergy medication, which is important because a lot of these medications take at least 24 or 48 hours to hit full efficacy. Now, for a lot of people who have allergies, and this is a lot of people, somewhere around 20–25% of Americans, for folks that have allergies, this merely makes them miserable, right? But there's a lot of people out there who have asthma or chronic obstructive pulmonary disorder, and for these people, exposure to pollen can be much more serious. So there's a real need for creating models of airborne pollen, and that is what a lot of my work focuses on. And so I do a few different things in order to generate these models. I make use of, I do a lot of field work. And I also use remote sensing, ranging from drones to satellites to try and understand things like where are the plants that are producing pollen, how much pollen is being produced, when is it being released, and ultimately, how does it disperse through the air? These are all of the ingredients necessary for creating process-based models of airborne pollen concentrations. And ultimately, this is what we need to do in order to get this information and predictions about pollen concentrations out to relevant stakeholders, as well as people with allergies who can use this information to improve their health. So I'm now going to take just a tiny bit of time to tell you about what this looks like in practice. And so some of this might involve things like using the color of these pollen cones for this species in Texas to try and scale up predictions of pollen production. This is Hannah, who's a graduate student in my lab and doing a fantastic job of trying to use these remote sensing indices to understand this at spatial scales ranging from meters up to site level, and eventually, as we incorporate more satellite data, to even larger scales. And so this is kind of an example of some of what's done in my lab. We also do a little bit of work on other things besides pollen. And so one of the topics that we're working on right now is trying to understand the cooling effects of trees a little bit better so that we can keep people out of the emergency department during heat waves. And this is some work that's so far mostly focusing on New York City. So these are a few examples, plenty of other ones, everything from identifying individual trees with remote sensing data to understanding correlations between urban tree composition and neighborhood demography of people, that is, things like income and race and ethnicity. And I also do a little bit of some other stuff, like creating novel devices, messing around with Arduinos, and trying to come up with the tools that I need in order to take the measurements that are required. So I suppose with that, I would also like to highlight some of the other folks that are in my lab with me doing this work, including Hannah Zonneville, Dave Miller, and several other students as well. And if any of this is interesting to you, come and stop by. We're up on the 10th floor of Bradfield. We'd love to chit chat with you all about any of these topics. Thanks.
So, any questions?
Do you think the pollen data can be used as an indicator of ecological health?
Oh, it's certainly useful for a lot of ecology. That's how I got into this. My background is plant ecology. And so it can be useful for understanding things like phenology, the timing of life history events. Ecosystem health for me, I'd say for me, I think about it a little bit more from the perspective of like particular types of plants. So it can tell you like, hey, are there a lot of plants of a particular type in a particular area? And so there's whole subfields around paleoecology that use pollen for that. So it can be quite a useful tool for particular things. Thanks.
I have a question. So at the state level, the New York state level, is there a very robust tension between those promoting ecosystem services and cooling of trees and then human health concerns? Or do those stakeholders not necessarily meet each other?
Oh, you get all sorts of interesting politics, right? Because there's a lot of people, including me, who think trees are great and we should have a lot of them in cities, right? There's $1.5 billion going into community and urban forestry right now through the Inflation Reduction Act. So we have a lot of people who care a lot about trees and want them. in cities. And so sometimes people get a little offended when you say, hey, they do bad things too. And so there are some folks who would rather ignore anything bad. For me, I think you really need to take both the good and the bad, weigh them all against each other to make good decisions.
So thanks for the invitation to speak with you all. I'm Ginny Moore, and I'm an assistant professor in plant breeding and genetics. And today I'll be giving an overview of my lab's work, which focuses on plant breeding for sustainable cropping systems. And we're housed in the plant breeding and genetics section, but excuse me, we also tread into the disciplines of agronomy and agroecology, integrating management and social science questions along with our genetics and breeding work. And before I start, I want to acknowledge the contributions of the staff and graduate students and many undergrads that have worked in my lab over the last 4 years since I started at Cornell. Our work is spanning disciplines, and we work on a lot of different crops and systems and do a lot of field work in all kinds of weather. So it's a really adaptable and hardy and hardworking bunch of folks that are working on these projects. So plant breeding for sustainable cropping systems, this encompasses a lot of different ideas and practices. And so this term, sustainable cropping systems, encompasses a lot of different ideas and practices. And I decided to structure my presentation today around the lens of soil health as a specific way to think about sustainable cropping systems, and thinking about breeding for characteristics of plants that keep this long-term goal of soil health in mind. And this framework that I have on the slide here, it was developed by USDA's Natural Resources Conservation Service, describing these principles that encompass a range of farming practices that maximize living roots, soil cover, and biodiversity while minimizing soil disturbance. And for plant breeders, we can approach working on these principles and improving these practices with genetics by either improving plant traits that support sustainability goals, or plant traits that support farmer adoption of sustainable practices. And I would say my lab, we do some of both, improving sustainability related traits directly, or thinking about farmer adoption, but we perhaps skew a little towards the latter approach, thinking about how we can make it easier for farmers to conduct practices that we already know enhance sustainability goals. And so I'm going to structure my presentation around these 4 principles of soil health. And the first one that I'll talk about is maximizing living roots, which one way that we do this in our lab is breeding perennial forage crops. So forages are especially important for New York State because of the prominence of New York's dairy industry. And we've seen, despite the many sustainability benefits that forages can provide, we've seen long-term declines in forage acreage in New York State and nationally. This has been consistent over many decades. And I think there's a lot of factors involved, economic and policy factors, challenges with managing forages. And some of these I think are a little outside of the scope of what plant breeding can address, but some of the management factors especially, I think breeding has a contribution to make. One of the main areas so this is, okay, so yeah, so acreage has been shrinking over the last over 100 years, or is down to levels below what they were 100 years ago. And one of the factors that has really contributed to management challenges in forages is plant establishment. So alfalfa seed can be pretty expensive, and farmers struggle to consistently establish stands that are uniform. And there's a few different factors that affect establishment. One of them is autotoxicity, so that's a form of self-allelopathy. So mature alfalfa plants produce compounds that suppress germination and growth of seedlings. And so it can be especially hard when you have a failed stand of alfalfa to then come in and replant. The compounds can stay in the soil and affect establishment for up to a year after a stand is terminated. And we've initiated a project looking at this, and so you can see examples here. We've in the top picture we have alfalfa seedlings that have been germinated in distilled water versus in an extract from alfalfa tissue, and you can see the effect that it has on germination and growth of those seedlings. And so one project that we're working on is developing alfalfa populations with high and low production of autotoxic compounds and high and low susceptibility, so that we can both understand the fundamental genetic mechanisms underlying this characteristic and develop varieties that are better able to establish following a previous alfalfa stand. And the next soil health principle that we work on is maximizing soil cover, and we approach this by breeding for overwintering cover crops. And I'm the director of the National Cover Crop Breeding Network, and this is a group of plant breeders and farmers and agronomists across the country who are seeking to develop new cover crop varieties that are better adapted to farmer needs and regional conditions. And we work on several different overwintering cover crops, including winter annual legume species, as well as cereal rye and several brassica species. And we're breeding for a number of different traits, primarily related to farmer adoption. So things like early vigor, ability to survive the winter in cold climates, and ability to produce a lot of biomass, which is correlated with a lot of the ecosystem service benefits that farmers are seeking. We also have lead the winter pea breeding program within our larger network. And one of the traits that we're specifically interested in winter pea is overwintering ability. It's not consistently overwintering in northern locations in the US and Canada. And we recently acquired a cold temperature growth chamber that will allow us to do more rapid evaluation of our pea germplasm and select populations that are better adapted to survive the winter in New York and in other locations across the country. And so we're screening for cold tolerance in a wide range of pea accessions. And this work is largely led by my PhD student, Raksha Thapa, who has really pioneered development of these protocols in our lab. And the third principle that we work on, maximizing biodiversity, we think about this largely through breeding for multi-crop systems, things like intercropping, breeding for mixtures of different species to grow well together. And one system that we work on is breeding alfalfa for intercropping with intermediate wheatgrass, which is a perennial grain crop. And so we're looking to develop alfalfa that can be a suitable companion for intermediate wheatgrass and for use in dual purpose systems producing both perennial grain and forage biomass. And so that's an example of row intercropping that we're pursuing in the lab. And then we also have a project focused on breeding red clover for intercropping with corn. And that's an example of relay intercropping. So we plant the corn first. So there's a period where it's a single crop system, and then we interseed red clover, and it has to survive under the corn canopy for several months before the corn is harvested, and then the red clover can continue growing and provide cover cropping and forage benefits for farmers. And then the final principle I wanted to discuss is minimizing disturbance. So we approach this by evaluating variety performance in reduced tillage systems. So As some of you might be familiar with, in organic cropping systems, farmers tend to rely on tillage as a tool to manage weeds rather than using herbicides. And this has a real significant trade-off in terms of soil health. And so there's interest in developing reduced tillage organic weed management systems, for example, using cover crops as a mulch to suppress weed growth. So this is an example of one of our technicians, Jesse Chavez, roller crimping a rye cover crop, or a rye, it looks like rye hairy vetch cover crop that will then plant a cash crop into directly. And so we've done some variety trials with a couple of different main crop species with hemp and with dry beans to evaluate variety adaptation to these reduced tillage cover crop based systems. And this can be a kind of challenging environment for cash crops. They need to emerge, germinate in cold soil and emerge through this thick layer of biomass that's, that the mulch comprises. And then depending on the cover crop that you're using, you might also have significant nitrogen tie-up. So it can be a challenging environment. So we're evaluating variation in different crop varieties to perform in these systems. And have also done some comparisons of different dry bean cultivars between tilled and no-till systems. And this is work that's been led by master's student Erica Everest in my lab. And it's also part of a larger organic dry bean project that some other folks in SIPS are involved with. And I will leave it there. Thanks, Josh.
Time for maybe one question.
Yeah, go ahead.
So with the alfalfa, the reason they make this compound and some other students from growing, do you guys have any idea? Because you said you have the extract. So do you have any idea what is in that extract? And like, can you relate that back to a gene or some sort of other system where they've looked at knocking these out or making resistant plants?
Sure. We're still learning what the compounds are. It's not been identified. There are multiple compounds that are producing this effect, and there is variation in both production and susceptibility in our alfalfa cultivars. And we are working with some folks at Michigan State to identify compounds and also look at interactions with soil microbiome. And yeah, it's a super complex system that we're still definitely unraveling. Yeah, great question.
I'm mostly going to talk a little bit about our flower bulb program that we had done for 25 years here. I'm mostly a greenhouse crops person, not vegetables, not edible food, but ornamentals. When we talk about greenhouse crops, and we've done a lot of work with flower bulbs and carbohydrates. So, bulbs are big bags of carbohydrates. So we were quite interested in learning more about that over the last, long time. And then I also teach the greenhouse management course, which we'll talk about that a little bit, which many of you participate in that, helping to teach that. Okay, all right. So we worked for 25 years with the Dutch Flower Bulb Export Association, which was sort of an interesting thing. They had a program that they started in 1965. And it was sort of a research/extension program to generate information that was useful for greenhouse producers in the US and Canada, and also landscape people, nurseries, anyone, commercially who was using flower bulbs. And so for them, they would support this research and some outreach, and presumably that would help the industry with their flower bulb problems, and they would buy more bulbs from the Dutch. So that was great. I was the second person to do that, and I think the last person. The first person was Gus de Hertog. He started at Michigan State and then ended up at North Carolina State. They published a book after 50 years, which was really quite nice. One of the nice things about this, we had a number of interns. We had 18 Dutch interns. The industry every year would send, practically hundreds of thousands of bulbs for experiments that we did at the greenhouse and in the landscape. And they would also send an intern to help plant everything and collect the data, and it was really great. We had, these were young students. Some of them were 21, most of them were not. The drinking age in Holland is certainly less than 21, and they carried that on whenever they got here in the US. We had open houses. We had all kinds of industry visitors. I only know of one arrest of these interns. That was really quite a funny story. I'd be happy to tell you about it, but maybe not here on the camera while things are being recorded. Okay, now, if I do this, this is great. Okay. So some of the things that we did, really the work was based on interesting problems in the greenhouse industry, okay, as far as forcing of flower bulbs, either as potted plants or as cut flowers. And so there were all kinds of cultivar trials of tulips, daffodils, and hydrangeas and lilies and all kinds of things. One of the issues with the production of greenhouse crops in pots is height control. And you might say, well, you could always breed dwarfer plants, and of course you can, but that's not always possible. So it's quite common to use anti-gibberellin growth retardants in greenhouse production of ornamental plants. And we did a lot of research on that. We did some interesting work with ethifon and realized that ethifon, which is a liquid, it's called liquid ethylene, 2-chloroethylphosphonic acid. It releases ethylene in plant tissue. At least in our tulips and daffodils, it crosses into the root and it moves freely upwards in the xylem, which was quite interesting. So we developed methods of applying ethifon as a soil application, which is quite unique for growth regulators. We did a lot of work on GA47, which turns out is a very potent inhibitor of leaf senescence in lilies, either as cut flowers or potted plants. Many of the lily cultivars have pretty atrocious leaf chlorosis towards the end of their production cycle. And the work that we did, and this was really Anil Ranwala when we first moved here from Clemson in 1998, Anil did a lot of great work, and it eventually expanded existing labels for GA47 to be used on lilies. And so Easter lilies that you buy at Easter time, if they have green leaves on them, you can be certain they were treated with GA47. If they have yellow leaves and they look bad, you can be certain that they were not treated with GA47. So practically, all of the lilies grown in North America are treated with this. We did work in these bud development meters. It becomes interesting in greenhouses when you're growing crops for a holiday period, and you have to have that plant flowering X number of days before the holiday so that you can sell it and get it into the stores in order for people to buy it. That is critical, not on a weekly cycle when you can have it go to market at any time. When you're growing for holidays, you've got, like Easter lilies, key example, you have to have these plants available at the right time. And so when you start looking at the temperature, the lighting, all the different things that you can do to make the plant flower on time, being able to predict, when the plant's going to flower is key. And with lilies, well, there's just gazillions of cultivars, and we spent a lot of time working on that. The trouble is the lily breeders breed more lilies than we really need. And so there's a lot of work there that, well, it's nice work, but bye-bye, because those cultivars all go away pretty quickly. I don't know, there's some interesting stuff about fusarium and ethylene. Fusarium produces ethylene and causes very severe problems on tulips especially when they're shipped in containers on vessels from Rotterdam to New Jersey. Interesting stuff about that. Simple things, planting depth. Something as simple as literally how deep you plant a bare root perennial is either leading to success or failure. That there's a huge bare root perennial industry in the Netherlands, and if these plants are planted like an inch or 2 inches deep in a pot, according to what the directions are, 80% of them will not grow. You know, they require a certain amount of light, certain stimulus to actually start growing, and very, very simple experiments, but quite useful experiments. Okay. So the thing that we've occupied ourselves for the last few years, because do I have a couple minutes? Okay. We have this planter. The Dutch sent us this planter with 100,000 bulbs, and they said, go plant some stuff. So it's a very interesting planter, and you would have seen, if you go downtown, you would have seen the results of this. The bioswale in the botanical garden. Here's the McClintock shed. This is down right near Boatyard Grill, Cass Park. We've planted about 500,000 daffodils downtown in the last 5 years that I think have been very nicely received. We've done research to support if you're planting bulbs in the grass, which is what this machine does, it lifts the sod, drops the bulbs under the sod, and then replaces the bulbs or replaces the sod as the tractor drives along. The question then becomes, when can you mow the grass? When can you mow the bulbs? Because we need to have foliage in order to build the bulb back up. So there's a conflict, and we've done work to figure that out. It's about June 10th. Downtown or down on Long Island, 75,000 bulbs, 3 hours, 3 people with this machine. So the companies have brought in about 15 of these machines in the last 3 years. And they've been pretty widely adapted throughout the Northeast and down into the South. So we've helped with that. And I see you're about ready to stand up, so I'm gonna stop. Okay? Thank you. Yeah.
I'm just wondering, are there any like elusive traits that you or other breeders have gone after for bulbs? That are just
Any what kind of traits?
that's really desirable but impossible to achieve, like
A black tulip.
a black.
It doesn't exist.
Okay.
There are very dark purple tulips, but no black tulips. One of the cool things, if you ever get a chance to go to Holland, there's a great, in the springtime, there's a garden up above Alkmaar, near Alkmaar, called the Horticulture. And it's a living collection of ancient tulip cultivars, including intentionally virus-infected plants, right? The whole, tulip mania and all of that. And there are cultivars, or there's tulip germplasm that coexists pretty happily with the virus. Usually tulip breaking virus is fatal after a few years, but some genetic backgrounds tolerate that widely. And they will have 3,000, of these infected tulips, all the same kind of color and pattern. It's astounding. It's astounding. So.
Okay, Bill.
Okay, yeah, thanks.
This has been a production of Cornell University, on the web at cornell.edu.